In this lesson, we will study bacterial cell internal structures. Cell membrane is delicate, thin and flexible structure, and it allows the movement of material or passage of metabolites, proteins and other substances, and it surrounds the bacterial cytoplasm, which actually contains cell internal structures. We can see here in this diagram that bacterial internal structure consists of this cytoplasm. Everything contained within cytoplasm is known as protoplast. So in cytoplasm we have plasmids, we have DNA and ribosomes, and in addition there are few other structures which we will see in the next slides. Talking about bacterial internal structure, we have nucleoid, which is actually DNA. It is a double-stranded helix and it is circular, and because it has a single nucleoid or single DNA, the bacterial chromosome is said to be haploid. In addition, we have plasmids within cytoplasm. They do not have any role in the growth and development, and they are actually involved in drug resistance or disease resistance, and they can replicate on their own too. Then bacterial cell cytoplasm has mesosomes, which are inward invaginations, and they have a role, or they are involved in the export of extracellular enzymes. They also contain respiratory enzymes. Actually, cell membrane also contains respiratory enzymes, and as these are inward invaginations of a cell membrane, so they also contain respiratory enzymes. Talking about the ribosomes, they are 70S in prokaryotes, and they are involved in the protein synthesis. And then bacterial cytoplasms contain granules and storage bodies like sulfur granules, starch granules. They are actually meant to withstand the competitive environment, the competitive conditions. Then they have bacterial cell have spores, which may be excreted out or secreted out, which are called as exospores, or they may be inside or known as endospores, and they are actually dormant structures, and they are formed on later stages of vegetative development, and they are actually present, or they are released because of to withstand the adverse conditions, adverse physical conditions like the fluctuations in temperature or the pH. On onset of favorable conditions, the spore germinate to form a vegetative cell. Then we have bacterial cysts. They are dormant, thick-walled structures, and they are formed later in differentiation of vegetative cells, and they are also formed due to the adverse conditions, and they can revert back once the conditions are settled.